Cultivation of Viruses, Part 2. In the previous lecture on cultivation of viruses, we learned about how bacteriophages can be cultivated within bacteria. In this lecture, we will learn about cultivating viruses in organisms such as animals and also in embryonated eggs. Viruses can also be cultivated within plants. But in this video, we will be examining this topic only with reference to animals. Viruses can also be cultivated in cell cultures, and you will study that in the succeeding video. Once again, to drive home the point that viruses are obligate intracellular parasites. They can only be cultivated within living cells. Laboratory animals can be used for routine cultivation of viruses, and some animals that are normally used for viral cultivation include mice, rabbits, monkey, guinea pig, and ferret. Mice are most commonly employed for such viral cultivation. The selected animal should be healthy and free from any disease. Suckling mice that are less than 48 hours old are commonly used for viral inoculation. Now, the virus can be inoculated into the animal nasally, which means through the nose, or intraperitoneally, which means into the peritoneal cavity, or subcutaneously, which means below the skin. So it all depends upon where you want the virus to go and which cells you want it to infect. After the animal is inoculated with the viral suspension, it will then be observed for signs of disease, for any changes in its morphology, for example, any visible lesions that it may show, etc. And once the symptoms are observed, the infected animal is then sacrificed to examine its tissues, and the virus can then be isolated and purified from the tissue. Now here you can see this is a diagram of a mouse which is being inoculated with a Zika virus. In this slide, you can see this. This Zika is actually a mosquito-borne virus. It's transmitted through Aedes mosquito. And there was an outbreak of Zika in Latin American countries like Brazil in 2015. And this was very much in the news because women infected with Zika during pregnancy gave birth to children with microcephaly, that is the head which was very small in size, and other congenital malformations. So in this diagram, Zika virus, in this example, it is inoculated into mice. Now, within the mouse, the virus will start destroying the cells. It moves in the blood to different organs. Now, viremia is a condition where you will find a concentration of virus in the blood itself, and the blood will take the virus to different parts of the body, to different organs in the body, and so the viral load, that is the concentration of virus in all these organs, it starts to increase. And ultimately, it will cause death. So the mouse here has been used as a model for understanding the Zika virus disease, and also for evaluating vaccines and antivirals. The organs of such a mouse would also be a source of the virus, which has replicated in good amounts. So the advantages of inoculating animals with the virus of interest are that we can determine what are the clinical symptoms of the disease, what, how does the disease progress, what is the pathogenesis, and also how can the disease be diagnosed. We can study the immune response, epidemiology, and also if it's an oncogenic virus causing cancer, we can study the process by which it causes cancer. We can also study the production of antibodies that are produced against the virus in this mammalian system. So these are the advantages of directly inoculating animals with the virus of interest. Inoculating animals with viruses has several disadvantages. It is expensive and maintenance is an issue. There is going to be both non-specific and specific response to the virus by the mammalian immune system. Now, every animal of the same species is not going to be reacting in the same way. So there'll be some variation in response to the virus. Individuals may show variation in response to the virus. And different viruses, they may grow in different animals. So the mouse may not be the correct model system for several viruses. And so we should know the correct host animal if we want to inoculate it and try to take advantage of it growing in that correct host animal. The host animal should also be easily available. Now, some human viruses cannot be grown in animals or can be grown, but they do not cause the disease there. And also, inoculation into the host animals could result in the generation of escape mutants. That is, the virus may mutate, or its genetic material may mutate so that it is able to defend itself from the host immune response. And finally, the inoculated animal, there may be some issues relating to animal welfare because the inoculated animal is going through a lot of pain and suffering as a result of this procedure.